Good morning, good afternoon, good night, or good morning again, everyone. Thank you for coming back to another educational video. This educational video is for all my IB biology students cramming to study for the test the night before. Do not use this as your only source of information. Today we will talk about, can you guess? DNA replication. You might ask, what is DNA replication? And possibly, what is DNA? DNA is a double helix strand that is composed of the stored genetic information of a cell, which can be replicated during, I just said earlier, DNA replication. DNA replication is a major component that makes you. A DNA strand is also anti-parallel. This means one strand goes at a 5' to 3' direction, and the other one is the opposite, so it goes from a 3' to 5' direction. To understand this, the prime is dependent on the direction of the ribose sugar molecule of the DNA, so it is dependent kind of on where the carbon is facing. DNA strands have 3 different groups, which includes a phosphate group, a deoxyribose group, and a nitrogenous base. There is a phosphodiester bond that is formed between the phosphate of one nucleotide and a pentose sugar. A covalent bond is also formed between parts of the nucleotide. A hydrogen bond is the bond between the nucleotide and or nitrogenous base pairs. This is an important bond because it is a very strong bond. Hydrogen bonds are very strong. You can learn more about that in chemistry. There are 4 different nitrogenous bases, which include adenine, thymine, guanine, and cytosine. These bases are very important because they code for your genetic information. DNA replication happens in the nucleus. Not all cells have nucleuses. This video will focus on eukaryotic cells, so cells that have nucleuses, not prokaryotic cells. DNA replication. Keep in mind that DNA replication happens before cell division, so it happens before mitosis and meiosis. DNA replication is a process by which DNA makes an identical copy of itself for cell division. So a little bit more about the DNA replication. So it happens during interphase, not helicase, is crucial. It unwinds the DNA strand to allow the DNA strand to be replicated. The helicase unwinds certain segments of the DNA, so portions of DNA that need to be replicated. So it places that need to be replicated. Yep. It is important to note that the enzyme helicase breaks down the hydrogen bonds that are formed between the nitrogenous base pairs to allow the DNA strand to become unwinded. You can think of this process as you unzipping your jacket. That might sound a little silly. Step is for the enzyme primase to go around and put a primer on certain regions of the existing strand to tell DNA polymerase, or you could call them DNA pol, where to start. DNA pol only reads the strand in a 5' to 3' direction, which we talked about earlier, about the direction. So it is able to read the leading strand continuously. In the lagging strand, it is a little bit more difficult. So they need a little bit more primer. In the lagging strand, it's a little bit more difficult. So it needs a little bit more primer to be put on. So it is easier for the DNA polymerase to understand where to go. Since the lagging strand goes from a 3' to 5' direction, where the nucleotides are kind of produced a little bit more discontinuously. DNA polymerase produces the complementary base pairs of the existing strand that is being read. Very important enzyme as well in DNA replication. So during the synthesis of lagging strands in DNA replication, there tends to be a lot of Okazaki fragments. Okazaki fragments are produced due to the lagging strand that is at a 3' to 5' direction. It is hard for DNA polymerase to read at a 3' to 5' direction, since it reads at a 5' to 3' direction. So a little bit more about this. The ligase assist in the bond formation of fragments. This is done through facilitating the joining of the DNA strands together by catalyzing the formation of a phosphodiester bond. At the end of all this, you will have two identical copies of the DNA strand. Each copy of the DNA strand will have a copy of the new DNA and a copy of the old DNA strand. This is what we call semi-conservative. This is why we say that DNA replication is semi-conservative. And thank you again, and enjoy this video. Hopefully you learned something, because y'all gotta study for your test so you don't fail. Thank you, and